Chapter 2: Characteristics of Cancer Cells. In this chapter, we'll examine how cancer cells differ from normal, healthy cells. Cancer cells possess distinct traits that enable them to grow uncontrollably, evade the immune system, and spread throughout the body. Understanding these biological changes is essential for grasping how tumors form and metastasize. This knowledge also provides insight into why cancer is so difficult to treat. One of the first signs of cancer is its ability to grow unchecked, even when crowded. Normal cells follow a rule known as density-dependent inhibition. When they sense enough neighboring cells, they stop dividing. Cancer cells ignore this rule and continue to multiply. This results in disorganized cell layers and tumor formation. It's one of the earliest and most visible markers of cellular transformation. Normal cells need to be anchored to a surface or neighboring tissue in order to grow and survive. When detached, they typically undergo programmed cell death. In contrast, cancer cells are able to grow while floating freely in liquid or soft tissue. This Anchorage-Independent Growth is a critical feature that enables metastasis. It allows cancer cells to survive in the bloodstream and invade new organs. Normal cells age and stop dividing after a set number of cycles due to telomere shortening. Telomeres are protective caps on the ends of chromosomes that shrink with each division. Cancer cells reactivate an enzyme called telomerase, which keeps these caps intact. As a result, the cells can divide indefinitely and become biologically immortal. This immortality gives cancer cells a profound advantage in sustaining tumor growth. Healthy cells rely on external growth signals to divide, ensuring that cell growth is well regulated. Cancer cells break free from this requirement by producing their own growth factors or altering their receptors to be constantly active. This gives them a self-sufficient growth signal that cannot be easily controlled. As a result, they grow faster and without the typical constraints that regulate normal tissue. This trait plays a major role in cancer progression. The cell cycle is a tightly monitored process in which cells grow, replicate DNA, and divide. In cancer cells, proteins that regulate the cycle, such as cyclins and cyclin-dependent kinases, are often overproduced. This pushes cells through the cell cycle without normal checks and balances. The result is uncontrolled cell division and a higher chance of mutations. These changes contribute significantly to tumor formation and growth. Checkpoints are essential for maintaining genomic integrity during cell division. They ensure that cells only divide when conditions are right and that any DNA damage is repaired. In cancer cells, these checkpoints, especially in G1, S, and G2 phases, are often bypassed. This allows cells with significant damage to keep dividing, accumulating even more errors. This unchecked progression leads to rapid tumor development and genetic chaos. Apoptosis, or programmed cell death, is a mechanism used to eliminate damaged cells. The p53 protein plays a critical role in initiating this process when cells experience DNA damage. In many cancers, p53 is either mutated or completely inactivated, meaning cells no longer receive the signal to self-destruct. This allows genetically unstable cells to survive and multiply. The loss of p53 is one of the most common and dangerous changes in cancer. The Bcl-2 protein works to prevent apoptosis and is often overexpressed in cancer cells. This overexpression allows cells with serious abnormalities to avoid death and continue dividing. As a result, tumors grow more resilient and harder to treat. Bcl-2 is especially relevant in cancers like leukemia and lymphoma. Therapies that target Bcl-2 aim to reactivate the cell's natural death program and shrink tumors. DNA repair systems are essential for correcting genetic errors caused by environmental stress or replication mistakes. In cancer cells, these systems are often impaired or non-functional. This leads to an accumulation of mutations that drive cancer progression. The inability to fix DNA damage results in a state of genetic instability. This condition accelerates tumor evolution and contributes to treatment resistance. Genetic instability is a hallmark of cancer and refers to the increased rate of mutations and chromosomal changes. Aneuploidy, the presence of an abnormal number of chromosomes, is a common outcome. These chromosomal abnormalities disrupt normal gene expression and fuel tumor diversity. As instability increases, cancer cells evolve rapidly and adapt to survive. This makes them harder to eliminate with a single treatment approach. The immune system is designed to detect and destroy abnormal cells, including cancer. However, cancer cells develop ways to avoid immune detection. They may reduce antigen presentation, produce immunosuppressive chemicals, or cloak themselves with surrounding cells. These strategies allow them to grow and spread without being attacked. Overcoming immune evasion is a central goal of immunotherapy. Tumor-specific antigens, or TSs, are unique to cancer cells, while tumor-associated antigens, or TAs, are found in both cancer and some normal tissues. TAs are typically overexpressed in cancer. These antigens are critical for developing targeted therapies and diagnostic tools. However, the variation between individuals and tumor types makes it difficult to create universal treatments. Continued research is needed to identify more reliable tumor markers. Cancer cells frequently alter their surface structures to support growth and movement. They reduce levels of E-cadherin, which normally helps cells stick together, and have fewer gap junctions for communication. These changes enable cancer cells to detach and migrate more easily. Surface proteins like lectins also increase, aiding invasion. These membrane alterations are central to cancer's ability to invade and metastasize. For continued growth, tumors need a blood supply. Cancer cells stimulate angiogenesis by releasing growth factors like VEGF. They also produce enzymes called proteases, which break down the surrounding tissue. This allows them to invade nearby areas and spread through the body. Angiogenesis and invasion are key steps in metastasis and are common targets for cancer therapy.